From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on March 6th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 82nd episode of Matters Microbial. Thanks as always for listening, viewing, and sharing the good microbial word. I mentioned in a recent podcast that I was going to show you all a little of what I do with students. In particular, I like to battle negative microbial PR by showing others how integral microbes are to everything. It is a microbial world. We just live in it. I am following 4 sites for this process. First is the water fountain you see here. I then swab from the water outlet onto R2A medium, which is good for water-associated microbes. As you can see, the resulting plate was interesting. In this close-up of that plate, you can see 3 basic types of bacterial colonies: white, orange, and reddish. I've streaked out each for further analysis. Are they characteristic of water? Our mouths? Something else? It's always exciting to find out. Next, I looked at our deionized water spigot as seen here on the right, which we use to make nutrient media, that is to say, just the water. The swab plate, again R2A, shows a robust microbial community, and the close-up shows its impressive diversity. Remember, this is just water and maybe dust from the air, and still life abounds. After that, I thought that looking at the water bottle refill stations seen here would be a good idea. The swab plate was interesting and exciting to me. In this close-up, it looks as if the larger white colonies are inhibiting other bacteria. And yes, I will be looking at this isolate carefully. Does it make antibiotics or other types of antimicrobials? Finally, near and dear to my heart, I decided to examine this small tree branch with lichens seen here. Just beautiful. The overall plate, after I dislodged everything from the branch and swabbed, was really quite interesting, as you can see. Look at the close-up with the diversity of pigmentation and morphology of the colonies. In particular, I enjoyed seeing 3 colonies that appeared to glow under ultraviolet light as seen in this video snippet. They may be making iron-sequestering siderophores, and those in turn may inhibit other microbes. Isolation? Checking for antibiosis, and identifying using 16S ribosomal RNA analysis. It'll be a fun journey to take you on and shows the power of working with students in a discovery mode. Episode 23 of this podcast featured Dr. Arturo Casadevall, who writes wonderful thought essays that I use every year when I teach, is also involved in science policy, and is an expert on fungal biology and pathogenesis. Last time, Arturo told us about fungi that could refrigerate themselves. We're lucky to have him back on the podcast to discuss how pigmentation can help fungi survive environmental challenges and how fungi are increasingly able to survive higher temperatures and why we should care about it. Welcome to the Quality Quorum once again, Arturo.
Thank you, Mark, and it's wonderful to hear that in your introduction you found pigmented colonies.
Oh, yeah.
And we can talk about the pigments. They are pretty, but they are doing something.
Yes, they are, and sometimes together in groups, which is interesting to me. So first off, I hope all is going well for you.
Things are well. I mean, with the ongoing uncertainty, but apart from that, trying to focus on the science and trying to manage our days.
I think a good place for our listeners and viewers to start is a reminder about, and this is my feeling that, and I don't mean to channel Rodney Dangerfield, but fungi don't get enough respect in terms of what I see being taught in institutions. So I think a reminder of why people should care about fungi is important. Especially, I don't know if you saw this, Arturo, there was on the 1st of March a wonderful article in the New York Times about fungal networks.
That's right.
And I'm going to put a link to that in the show notes.
Right. Fungal networks under your foot when you walk on the forest. Little did you know that under your feet there is a vast connection of hypha exchanging information between trees. And this is a world that we're just beginning to discover. We didn't know it was there.
And last time that you were on the show, you talked about the fungal takeover of the ancient world with the giant fungal fossils. And it's not simply the organisms that cause disease, nor is it the fruiting bodies that you and I call mushrooms that we slice onto our salads. Fungi are everywhere, and they do lots of different stuff.
That's right. And unfortunately, for reasons that I don't understand, but maybe it has to do with the fact that they are largely unseen, we tend to be largely oblivious to the fungal world, except when it intrudes in ours, like when it wrecks homes after Katrina, or when people get disease, or when it affects toenails, or when it's the heat of the summer and they cause athlete's foot. But apart from that, it's remarkably how oblivious we are, including in microbiology courses. You know, mycology may get an afternoon.
Yes. I don't have a solution for it other than multiple classes.
Yeah, well, I'm sure that works. And you know, just talking about it, I think the more people learn about it, the more they become aware of this incredible kingdom that is so essential for life on Earth, because it basically recycles all our nutrients. It's the major recyclers out
there. I've seen articles about them actually solubilizing minerals and bringing them to the surface that allow vascular plants to grow.
That's right. And, you know, fungi are now being developed that can eat plastics. I mean, that is going to be really important given the accumulation of this essentially non-destructible materials that are accumulating all over the biosphere.
So this is a great topic in general, and you're making me think that education should be focused on the microbial, because then we would be able to cover all the different parts of the microbial world, kind of avoiding, well, I'm going to allow fungi to be called eukaryotes because they are, but they're very unusual eukaryotes. And so as a result of that, I think it's important, and I really like what you said, I really do, that we have a focus on things that cause disease. And as you and I have talked about before, it's all about context. So it isn't a matter
of location, as you often put out in social media.
Exactly so. Exactly so. And that's why when you or I talk about microbiology with the public, the automatic response that people have is negative to all these germs of whatever form. And it's just not true, because after all, they were there first.
Absolutely. And when you think about the fungi giving us penicillins, they're giving us other antibiotics, they're giving us the statins that a lot of people take, they're giving us the cyclosporin, which is revolutionized transplantation, and they also have given us LSD.
Yeah.
I mean, the question is, why would a mushroom want to make something to get a mammal to hallucinate? And I don't know the answer to that, Mark, except maybe if you hallucinate and you have a bad trip, maybe you don't eat the same mushroom again.
There's something that's ascribed to Leslie Orgel or Francis Crick, depending on who you talk to, that evolution is smarter than you are.
I agree with that.
And it's I just think that we're so inwardly directed, it's hard to keep track of things. So let's move to today's topic, because you're really you sent me 3 papers that I think are quite remarkable, and I'm going to provide links to them, and hopefully we'll see some of the figures in this presentation as well. And I mean, the one that fascinates me first off is the relationship between pigmentation and the ability to survive in the cold, which is kind of the reverse
That's
of
right.
what we talked about last time.
That's right. I mean, Mark is talking about an article that was published in Current Biology. First author is Radamés Cordero, and as always, he gets all the credit. In which I would point. So the idea when we started this, we thought, melanin protects against sunlight. So you would think that maybe the organisms, if you look at it as a global distribution, the organisms near the equator will be more pigmented than the organisms in the poles. And what we found out was the hypothesis was completely wrong, that it was exactly the other way around. And when you think about it, it makes sense, because it turns out that melanin is this remarkable pigment when it comes to capturing electromagnetic radiation. So if you are pigmented near the equator, you will overheat. But if you happen to be in a mountain in Antarctica, that pigment could give you just enough to keep going.
And I want to be clear, you're not just talking about heat.
That's right. I'm talking about the whole electromagnetic radiation. The absorbance spectrum of melanin is remarkable in that it is monotonic. What it means is it absorbs everything from from the infrared all the way into the far UV. And in fact, many years ago, we showed that it helped interact even with gamma rays, and that some of the fungi used melanin to capture energy. That was something that was inspired from the finding that the mushrooms in Chernobyl, not the mushrooms, the walls of Chernobyl are black because there is a fungus growing on it that makes melanin.
You're going to laugh a little bit, but when I have first-year students, sadly no longer teaching first-year students, but I would ask them why plants are green. And I want them to tell me how we only see the reflected light.
That's right.
And that tells us something about how photosynthesis doesn't particularly care for green wavelengths, although there are exceptions. And I would make a joke, which is relevant to today, about an extraterrestrial organism that used more of the wavelengths. What color would it be? And I'm leading them to black.
Exactly. Black is, the melanin is just remarkable. It is so complex. It's composed of, it's not a protein. People think it's a protein or something. No. It's a cross-linked indoles and phenolic groups. And what happens is it is so complex that you can find a resonance for practically any wavelength. So things that fall into it get absorbed. Also, it is a stable free radical. Think about it. It's a biological stable free radical. You can take it out. You can put it in an electron spin resonance machine, and it's got a huge signal. And when light falls on it, it basically kicks out even more free electrons. Here is something, Mark. The structure of melanin remains unsolved. And the reason it is unsolved is because our science is wonderful at solving the structures of things that can go into solution or that can be crystallized. But it can't deal with substances that are amorphous. So it has local structure, but it's disordered. It's not that different than wood. We don't have the structure of wood. We think it's lignin, cellulose, or something, but nobody can write it up, can put up, what this table is made out of.
Oh, that is so interesting. I'll put links to some of this because I was thinking to myself, I know how cytochromes and chlorophyll work, and there's double bonds next to single bonds that tend to pick up energy pretty easily. And so I had assumed that we'd see a lot of that in melanin. Maybe we do.
So actually, Mark, you're right. So what happens with all these pigments is that they have a lot of double bonds that are near each other, such that they can interact and basically begin to absorb energy in the visible and even in the infrared. Imagine that instead of just a protein or a pigment in which you have only a few double bonds, that you have a structure in which there are zillions of molecular moieties that are cross-linked. And it allows essentially a sea of electrons That is why we can easily measure it to be a stable free radical. The electrons are delocalized. So my view is that melanin can any photon that falls into melanin can find some place to be absorbed.
And when you say photons, that covers things that people generally don't think about, including gamma radiation.
That's right. Gamma radiation. You know, when this work was done with Katerina Darachova, who's now at the University of Saskatchewan, and early on, she showed in the paper that the gamma radiation promoted the growth of melanized organisms. And people came up to me and said, Arturo, X-rays go through everything. What mechanism do you have for gamma rays being absorbed? And I remember being very upset at that, because that was a real scientific criticism that I didn't have an answer for. But we worked for many years, and we were able to show, for example, that if you make a melanin electrode, and you shine gamma rays through it, you get a current. Now, that's published in a single-word journal called Electrochemistry. But
that's amazing.
Exactly. That basically, there is no way out. If you make a melanin electrode and you turn the light on and you get a current, then the light at gamma rays, then you need to have some sort of interaction between the gamma rays and the melanin. Now, some people have speculated that it may be polarization, that these very energetic fluxes polarized it. But in some way, it is interacting. So you have biological matter interacting with very high energy electromagnetic radiation.
I think it was Zent Yorgi who said that life is an electron trying to find a place to rest. And I mean, any type of maxim is overstated. That's in the nature of things. But it's just amazing to think about this, because I'm a big science fiction reader, always have been. So these ideas where, nothing can survive around gamma radiation, right? Guess what? Some organisms can not only survive, but benefit from it.
I mean, these were very difficult experiments to do back then, because in contrast to chlorophyll or to plants, where you can just show it by putting them in the dark, they wilt. The fungi have all these enzymes, and they could break down agar. So you are always looking not on an all or none phenomenon, but a quantitative difference. That is, when you put the melanized in on the radiation, they grew faster than the non-melanized. And it wasn't, and people say, well, it was damage, it was something. Well, Kate was able to show that if you take just isolated melanin, and you irradiate it with gamma rays, and then you add NAD, that it can reduce it to NADH.
Oh my goodness!
So that part isn't there. So you can go from high energy electromagnetic radiation to reducing a component of metabolism, and that's all sitting in that paper. That paper, by the way, had a great odyssey. It spent 1 year at a single-word journal. And at the very end, they wouldn't publish it. So we said, well, we almost made it. Let's go to the next single-word journal. So then we went to the next single-word journal. They made us do a lot more work. And again, a year later, it wasn't. And then PLOS Biology had just come out. And I remember looking at the instructions for PLOS Biology and saying, we just care what the data is good. We're not going to make any judgments about importance or anything. So we sent it them. They gave us more work, and it was published in 2007. It was their lead year. And it's
when most people hear about melanin, Arturo, they tend to think about the pigment in our own skin. But from what you've told me, melanin is very widely distributed. So could you comment upon that issue and how it relates to the evolution of melanin?
So melanins, as I mentioned to you, are structurally very complex, but they are very easy to make. They often require a single enzyme and a substrate. For example, in the fungus, they use phenol oxidase. In our skin, we have tyrosinase. So we have a paradox. We have a very complex molecule that can do a lot of things, but it's very cheap to make. And it is made by all life forms, plants, fungi, bacteria, protists. Even and the only one that I don't think makes it are viruses. But you got to figure that this thing was invented or stumbled upon early in life. And life has taken this pigment and used it in a lot of ways.
Prior to the ozone layer, Arturo, we got a lot of UV light on this planet. So I'm wondering whether melanization could have been advantageous to early life forms.
So it is so easy to make that I'm a believer that it was there very early. It may have been in the before the biotic soup. And if I live long enough and I get to do, I would like to do some origin of life type of experiments using melanin, because I cannot believe that very complex surface will not catalyze all kinds of reactions.
So in this first paper, you were able to show that if you had melanin producing organisms, they grew faster at colder temperatures. And you're just measuring this in terms of growth rates?
No, that was not in that first paper. That was only looking at gamma rays. It's the 2017 the current biology paper. It's either 18 or 17, in which Radamés is able to show that if the melanized organisms in the cold room grow faster because they're using a little bit of energy from the light.
It's so interesting to me because you think, and forgive me this, you think, oh, it's dark, it's going to pick up heat, right? And it's not as simple as that, because there's actually an energetic advantage, which is just amazing to me.
So I think that this is it's a remarkable area. People have asked me, have you done more? And I said, we do a little bit, but we have never been able to get this funded. Why? Because it's a type of science that falls in the interfaces. Nobody owns it. And that's a structural problem with the way that we organize our science. A great discovery is made at the interfaces, but the interfaces don't belong to anybody. And when you're dealing with a review group or something, they basically review their areas and protect their areas.
Oh, I mean, I have said over and over again on the podcast, when you bring people together from different areas, that's where the excitement happens, because nobody knows what a bad question is.
That's right.
Yeah, because there actually aren't. I said this recently, that there aren't any, when you're wrong about something, it's not a problem, it's an opportunity.
That's right. There are no stupid questions. There are no bad questions.
Right.
The only question.
There are unprepared questions, but that's not stupid.
I mean, I think the only difference is there are questions you could answer and questions you can't answer. But
yeah. So I love the fact that you've shown this relationship, and I want to emphasize to listeners and viewers, it's not simply about the dark coloring absorbing heat. There's an energetic advantage to the organisms that helps them survive better in the cold. And again, that reversal that you told us of what we might expect, where you see more pigmented organisms toward the equator, less pigmented toward the poles that we're familiar with when we look at animals.
No, it's the other way around. That's what we thought. We thought the pigments will be in the equator.
Right.
And then we were wrong. And that's because the pigments overheat them.
And that's where I was going, and that's what I was trying to say. It's reversed from that, and it's really interesting. But they overheat if you take a pigmented microbe versus a pigmented fungal isolate and a non-pigmented one, and you have them at a given temperature that's higher.
Yeah, they will overheat. I mean, in fact, in that paper it shows how rapidly the temperature rises for melanin. It will increase several degrees centigrade very rapidly. So it's pigments. We have to start really thinking about pigments and what the microbes are doing now. The funny thing is, you began this section by talking about the pigments that you found in water. Now, if you had a single isolated organism, you don't see it, right? You don't see it in the microscope. But once you get a colony, then there is sufficient, absorption for you to see it. But that pigment is working at the single level organism, even though for you to see it, you need to see the colony.
And so many questions. My head is exploding about making amorphous compounds, how microbes can do that. And it's the same thing when you talk about lignin in plants, right?
Yeah. I mean, these are limits to our science. And the interesting thing is because there are limits to science, then people don't work on them.
Eh, well,
You
I
know,
mean, without being topical, it's hard to get funding for really new
right.
ideas.
That's right. But I think that, a lot of the problem is a lot of the stuff in nature is not amenable to our structural analysis. I mean, a good example is the cell wall. It's, I've written it is not a wall. I mean, and yet it's composed of multiple components coming together. It can move from so, but our analytical techniques fail to give
And
us a clear picture of it.
it's important to remember that the words we use to describe things, we're used to this in sociology. The words we use to describe things affect the way we think about it, but it's true in science too.
That's
And
right.
that's a really great example, the cell wall, because I've had students get really confused about that. Like, well, it's not really a wall then, is it? And I said, well, more like a series of networks. And then I try and get flippant with them about stuff. But it's really interesting because we can't put our perceptions on that level.
That's right. Neil Gao and I wrote an essay maybe a year or two ago on the cell wall metaphor and how much it's hindered science. I mean, one good example of that was that people did not believe that cell-walled organisms could make extracellular vesicles. They can't go through the wall. You got a wall. You can't get through. Or the vesicles come from gram negatives because they have another layer outside. And yet, when we found them in the fungi, I mean, it was just people looked at us and said, something has to be wrong. Maybe the lipids reassembled themselves. No, they're there. And then we went to find them in the gram-positive bacteria. And today they're accepted. But the problem is, it's the word. When you tell people that there is a wall, people think that things can't get through. And we don't have a physical metaphor for what this cell wall really is like, because it's got to be flexible. Things have to get through it. It's got to repair itself. We don't have anything in our macroscopic world that you can call it.
In my early days when I was studying the predatory bacterium Delavibrio, and I happened to meet the late, great Terry Beveridge, who was a wonderful electron microscopist, and he was very nice to me. And I made the mistake of saying periplasmic space to him, and he literally slapped my hand. and he said, it's not a space. It has a gel-like consistency. It's full of solutes and enzymes. And this is exactly what you're saying, that the name that we give something influences how we think of it.
Well, think about the word pathogen.
Yeah. And so it's a plasm.
Exactly.
It's not a space. Oh,
Yeah.
I mean, that's off some of the papers you wrote, but my goodness, every time I talk to you there, I have a series of notes here about things that you've said. Thank you.
Mark, you're very kind.
And the thing about and I'm going to put a link about extracellular vesicles, because that's a really big deal in microbial biology now. And, people initially say it's not true. They say it's true a little bit, but it's not really important.
But it's
and
not
then
relevant.
they say it was a good idea
true.
all along.
And then there's a great quote that I have in one of my papers with Ferric Fang. I think it goes like this. First they say it's not true. Then they say it's true but not important. And they say it's true and important but not new.
And they found it first.
Right.
That's the other one.
So at every level you encounter problems, and we should just embrace this is the way science is,
Well, the advantage to science is that you can come up with ways to establish the validity of what you're saying.
That's right.
And that's what I love about science right there. So this is very cool. And that's a joke, isn't it? It's actually cool but hot is what we're talking about. And then you went on to and all of this is based on the idea, this hypothesis that was put together about 2018, as you mentioned. But this takes you to some really interesting issues. And I want you to tell listeners and viewers why we should care about temperature and fungi, just in case they don't know.
So the fungal world, as far as we know, is hypothermic. That means that they prefer to grow at temperatures cooler than the environment. And their metabolism is adapted to temperatures that are cooler. But now the fungal world is about to have a huge problem. The world is getting warmer. And the fungi are going to have really two choices: to go extinct or to adapt. And the big concern is that we are protected against the fungal kingdom. The reason most people don't worry about dying from fungal disease is because immunologically intact humans and mammals have tremendous protection against the fungi by being 37 degrees. The problem arises that most fungal species cannot grow at 37 degrees, and yet many of them are pathogenic for plants, insects, etc. Guess what may happen if they adapted to be able to survive global warming? Then we could be seeing a whole new class of pathogens that we don't know right now.
And this is, again, it takes me back to science fiction, because when I was a boy and I would read science fiction, and before they knew what the surface of Venus was like, the idea was it was this very jungly place, and the first explorers were attacked by fungal diseases that took place in days. And I remember being like, wow, but that's not exactly what we're saying here. But the fact is, we're protected because of our elevated body temperature. Right. And most people who keep, say, tropical fish or certain reptiles are very aware of this, because there are lots of fungal diseases that go after reptiles, amphibians, fish. But we're pretty safe from it. But we know a lot because of immunocompromised individuals.
As you probably know, about 20 years ago, I had a lot of fun proposing. One of the things that really has animated me is I don't understand how such an energetically demanding life form like mammals and birds have become the dominant animals after the KT boundary. And it turns out that you can find massive fungal proliferation at the KT boundary. Whenever the earth has a bad day, the fungi do well. And imagine in that post-apocalyptic world, after the asteroid hit and the blast killed the dinosaurs or something, you had small mammals and you had small reptiles. How come we didn't get a second reptilian age?
Yeah.
The argument that with a lot of fun proposed is that the fungi kept down the reptiles, and then the mammals did well. And then when the skies cleared, there were a lot more mammals than reptiles, and the mammals went on to these large populations began to give us everything from whales to humans to monkeys to bats.
Birds, though, are an interesting subject.
Birds are a different ballgame, but there you may need the temperature right for high metabolism, because their energy, since the efficiency of the energy is dependent on the efficiency of the engine is dependent on the higher temperature. But the only two things that make it out of the KT boundary and do really well are hot, and those are mammals and birds.
That's really crazy to think about. Much as we love the idea of intelligent dinosaurs, right? Still.
Well, people mistake the hypothesis by saying Arturo says that the fungi killed the dinosaurs. No, that's not what I say. What I say is the dinosaurs died because of the calamity. They were at the top of the food chain. What I'm interested in is why, when you travel around the countryside, what are the large animals that you see? You'll see mammals and you see birds. You don't see any large reptiles.
And I think it makes sense. And again, that's just because of the perturbations in the ecological system. And I'm not even talking about the even stranger extinctions in the further past. This has been really fascinating. And if we're concerned about the issue of global warming, I think that leads us to a really interesting concept from your preprint, which has to do with heat islands. Could you tell listeners and viewers
So
about it?
first, I'm going to give 100% of the credit to Daniel Smith. He's a postdoc in our lab, and he has pursued this, and he is the one who developed this, the candy. Uh, what's the name of the candy again? Starburst.
Starbursts.
Starburst thing. So look, when we live in cities, cities are our constructions. They are warmer than the countryside by as much as 5 to 8 degrees centigrade. The reason for that is, think about all the concrete. We got rid of the trees. We got rid of a lot of the things that shield us. And the idea was, so cities are known to be heat islands. So the question is, could these heat islands be essentially places where you have much more rapid thermal evolution? Because the sidewalk is getting up to 140 degrees Fahrenheit. So anything in there is going to be under tremendous amount of thermal selection. So what Daniel did is he first developed the starburst technique. What's the starburst technique? He went and got himself candy. And how do you pick fungi or microbes off the sidewalk? Well, he figured out you take the starburst and you just press it on the sidewalk and bring it back to the lab, and then just put in a solution and plate this stuff. So, and then he's been looking at the fungi in the cooler neighborhoods and the warmer neighborhoods. So we are within the heat island of Baltimore. And guess what he can find? He can find that the ones in the cold, in the warmer neighborhoods are losing their pigment. Why? Because if you're making melanin, if you're making any of those dark pigments, you're going to get a lot warmer. And very interesting, when you find the same species in a cool neighborhood or a hot neighborhood, and you take them back to the lab and you put them in something called a heat ramp, which is a thermal torture machine where you basically put the microbe and turn on the heat, that the ones from the warmer neighborhood are much more resistant to the heat ramp. So this is happening. It's happening within our cities. And the big concern is that a lot of this stuff out there on the sidewalk has pathogenic potential. In fact, some of the ones that he's pulled out are opportunistic pathogens for immunocompromised people. But, it's got to be happening to all the fungal species in the environment.
So I have a saying that the hand of Darwin is on all that lives. And even in my own little lab, I can do evolution experiments, say by selecting for microbes that swim faster than other microbes.
That's right.
And it's easy to do. Students love it. It's really fun. But then you think to yourself, are there environments on the planet where this is happening naturally? And you've basically said for what's the term? Anthropogenic? Well, anthropogenic?
Anthropogenic, yeah.
Right. That we're doing it is the point.
And we've been doing making wine, fermenting. I mean, why Saccharomyces can take a lot of heat.
Yeah. So this idea about evolution taking place all the time, and about optimizing the ability to grow and survive and reproduce is really critical. And so we have a situation here in these heat islands. And by the way, the idea, I have to laugh about the candies because they're nice and sticky. We all know this.
Right.
And I've always worried about swabs. I've always worried about swabs not picking up everything. And how would you know, right?
Right. It's proving a negative.
At least we know that the candy's sticky.
Sticky. And this was entirely his idea. My only contribution to it was, so we are getting close to submitting the paper. And I finally said to him, so I said, Daniel, why don't you just make sure and check that the candy are sterile, right? Before you, because we just make sure there isn't something in there. And he checked it and they are sterile. So I guess anything that is soft, sticky, non-toxic could provide a great tool for picking up microbes from surfaces, which is actually very hard.
And this is interesting because many people will just do a swab of an environment and then do, in your case, this would be ITS sequencing to figure out who goes there. But this way is superior because
First of all, I don't know if it's superior. I'm humble about this. All I can say is that he developed this, and he's been able to pull out the fungi from the different neighborhoods, and he's been able to find the thermal differences. So I think we have a method. We didn't compare it to walking around swabbing rocks or anything like that, or sidewalks. But it is, I think people that are interested in environmental sampling should consider that there is a relatively inexpensive candy that could be used on surfaces that for sampling. Whether I imagine this works on tree bark, it probably works on rocks, it works in everything.
Well, I like the idea because if you do swabbing, and again, I want to be very cautious in what I say. I want to follow up on what you just said about humility. You can. It's sometimes difficult to determine prevalence.
That's right.
Because when you're using PCR-based techniques, there are ways to do it. I know there are. But so if you're doing that survey, as it were, is it really a survey? What you're doing are things that you can cultivate.
That's right.
Someone can argue that that is a subset of what's there, and they're probably right. But at least you know that what you got, you can grow.
That's right. And, it's not in that preprint because he's got a lot more data and we need to update the preprint. But it is interesting that on the warmer areas, the fauna that he pulls out is reduced. So in other words, you already beginning to have heating, reducing fungal diversity in the warmer areas. So and this is happening, I bet you it can happen in Tacoma if you did the same experiments there. And, to our friends, the bacteriologists, I would be thinking that the same thing is probably happening with bacteria, and it's happening with other organisms.
Do you know what types of, I'm going to say mutations because they are. Do you know what kinds of mutations would give thermotolerance?
Yes. Well, no. Do I know? No. But have people looked at this? Yes, and they have looked at it primarily in Saccharomyces. And here is the most interesting thing, Mark. You don't need many changes. I used to think that to make something more thermotolerant, you need to make the proteins more stable and all that. So when they sequence these strains, because people have done adaptive evolution in the laboratory, you can do it. Take a plate and raise the temperature of the incubator and come back in a week and see what grows, and then plate it again and raise the temperature again. And what you find is that there aren't that many. So it seems that it's probably a general tuning up of things. You're probably tuning up, you're creating epigenetic changes. You're tuning up the elimination of proteins that are damaged. You may be changing things that are not genetic. For example, lipid composition, things like that,
yes.
that wouldn't necessarily be picked up very easily. But I think the take-home message, Mark, is that this is not hard to do. And if one can do it in the lab and it can happen on the sidewalks, life has got a lot of potential out there for handling these extremes.
So I have a question, and it's one I don't know the answer to, and I'm embarrassed that I don't. Horizontal gene transfer among the fungi.
So there is, it can happen. In fact, in Cryptococcus, Joe Heidman's lab has shown that you should get him on the program sometime. That it appears that even bacterial genes have ended up in fungal genome. So horizontal gene transfer is happening.
So the reason here that these heat islands could conceivably be a source of genetic innovation that could be for thermotolerance that other fungi could use more easily than having to come up with those mutations themselves.
That's right. And here is another wrinkle to this, Mark. Another person you may want to have in the program, Asiya Gusa. She is a Duke. And what she's shown is that whenever you raise the temperature, the transposons hop.
Yes.
they hop in this fungus. And I'm sure the same thing is happening in bacteria. And when they hop, a lot of the jumps are detrimental. But an occasional one gives you drug resistance. An occasional one increases virulence. So here you have yet another element, which is that the mutation rate increases with temperature. So we're going to have adaptation. We're going to have potentially the emergence of traits that were not part of the original organism.
It's amazing.
Yeah, she's done beautiful work, and I strongly recommend her as another one of your guests.
Oh no, I would love to, because one of the things that happens is that I'll have people on the program who I've met and I know a little bit from my interest in microbial diversity. I want to have fungal biologists on, even though they're eukaryotes.
Well,
But come on, there's so many cool fungi out there. There really are. And then, of course, all the viral people, that's really important to talk about as well, because it is a microbial issue.
Absolutely. And, I'm a generalist, Mark. I see continuity. I don't see differences. I look at my stuff and I look at the bacteriology. I see continuities. I think one of the problems that we have in microbiology is that we encourage being partisan, and in reality, it's the continuity and the similarities that often are far more instructive than when you narrowly focus in on one organism.
I think that's a really good way to look at it. And, I can be arch and say about the microbial world, first evolved, last extinct, and that's true. But it is also true that some very interesting things that have been seen in bacteria and archaea occur in eukaryotes.
Right.
And then we often find things of interest in eukaryotes have analogs in bacteria and archaea. So this, once again, we can learn from the different systems. And in a perfect world, there'd be a research institute where you have all these different types of people together, but we tend to silo ourselves.
That's right.
Because we want people just like ourselves.
Yeah, and it's just the structure of our science. It's always a problem. As knowledge increases, people specialize. As they specialize, they basically silo, and that tension between the great advantages of generalism versus the great advantages of being a specialist. It's a real tug of war. It's a real tug of war, and you're not rewarded in this world for being a generalist.
No.
People are a lot more comfortable if they can say, Mark focuses on this organism, and that's what he works.
That's right.
If he basically tells you about a bacteria and then tells you about a fungus and then tells you about it, people say, I don't know what he's doing.
So I don't know if you've heard about the fungus Podospora anserina.
Yes.
But it has a lifespan. And this is a really confusing thing. And I made the mistake of doing that in my first year of graduate school, where I talked about it as a seminar. This was not received well by some of the microbiologists there, even though it was in the journal Cell. And what happened is a segment of the mitochondrial genome would amplify, and these organisms were obligately aerobic, and that was responsible for their lifespan, which didn't involve sporulation. And there was a really wonderful fungal biologist at where I was in graduate school named David Perkins, and he had known Beadle and Tatum. And so I'll never forget, he's sitting here carving up an apple as I'm trying to give my talk, and I keep getting interrupted by all these famous scientists, so I'm like shuddering. And finally, without even looking up, he says, "Stop. It has a lifespan. It's been known since the 1930s. Please go on, Mark."
In other words, the other people were giving you a hard time.
Oh.
And he basically says, stop it.
Did you notice I didn't name names? So
no, very political.
In any event, what's important there is that you need to bring lots of different ideas together for that purpose. He knew about that. I am not surprised by any complexities among fungi because I did a little work with Neurospora and its circadian rhythm
Right.
in race tubes.
That's right.
They can see light. They can have a clock. They can set it. Turns out bacteria can do that too. And this is because you have to have a curious mind.
Well, you're a generalist, Mark,
I am.
and so am I. And
Yeah.
I learned to. I'm curious about everything, but I also learn how to write really, really narrow grants because I know that in our world you get funded through that mechanism, but never lose your curiosity.
No, you can't. And I understand the directionality. I mean, for example, let's just say you're working on issues that have direct relevance to medicine. You're going to get all this pressure to really focus. And I understand that. I really do. But there are other ways of looking at science as well.
So, Mark, you made the off comment earlier in our discussion about microbes in Venus before we knew that it was boiling.
One more time, Arturo.
You made the comment earlier in our discussion about Venus
Yes.
and potential, but we now know that it's boiling. But you know what? It would be a really good exercise to sit down and write what would a microbe need to survive in the clouds?
Yes.
What would you put in its vacuole so it will float?
Well, that has to do with the density of the atmosphere.
Exactly.
Right.
And what pigments would you put in so that it can use the CO2 and things like that? Fix the CO2, and you can probably do it.
And I do not know how liquid droplets are colonized by microbes in our own atmosphere. That was something I'd have to learn about because there's certainly liquid droplets there.
But I mean, that'd be a great exercise to do.
Yeah.
And you know what? It may one day have practical applications if you want to terraform the planet.
Well, I mean, any place that there's liquid water and any type of energy differential, microbes can eke out some kind of living. And my money is on the microbes.
Absolutely.
Yeah. So do you have some take-home lessons for listeners and viewers from today's session that you'd like them to remember? The first is, of course, specialization is for insects. I've got that.
No, a specialization is really important because
yes, it is.
To make contributions, you need to go deep. Yeah. But on the other hand, the problem arises that you don't want a specialization run amok. You want to always retain, why did you go into science? I think we all went into science because we were curious and we were interested kind of in the big picture. And to hold on to that, even though there is this tremendous pull, to the area, to the comfort zone of specialization. But, I think that the topic of this podcast is pigments. I think people should look at their microbes and wonder whether they're white or whether they're pigmented, and think about what that pigment may be doing in the biology of the organism. And think that even though you can only see it when it's a colony, that pigment is there when it's a single cell.
I know that some pigments that microbes make have antimicrobial properties, like violacine in chromobacters and genithobacters. I've always heard, and I have not done this experiment, but I've always heard that the yellow color in Micrococcus is beta-carotenes for UV protection, and now you make me suspicious of that.
But it may be that the beta-carotenes are there for energy harvest.
Yeah.
To do the experiment. And, these are not easy experiments because even if you take a one with and the other one without, you got to rule out all these exogenous variables in growth and things like that.
I mean, if you think about it, I'm sure you saw this report. When very hot water at a hydrothermal vent hits the cold water, you get some triboluminescence. You get a very small amount of light that's generating. I don't know if you know about wintergreen lifesavers, but if you crush them, you get triboluminescence. Turns out there are cyanobacteria that can harvest that near-infrared light.
That's right. And think about it, harvesting near-infrared light. Those are photons with not a lot of energy compared to the stuff that's falling on a green plant. And yet life finds a way to use it.
Very interesting stuff. Arturo, I want to thank you for this wonderful opportunity to chat
And
about
thank you for
stuff.
having me back. It's always a pleasure talking to you. You're a generalist, and one can talk about practically anything.
Well, and I have several pages of notes of things that you've said that give me things to think about. And I think that's really important. And I hope it's been valuable for listeners and viewers. I mean, I make a joke that a lot of this podcast is for my personal therapy, but I also really want to share my love of the microbial sciences by having folks like you that talk about their career, their experiences. And what's interesting here are several themes that you covered. First is your absolute insistence on giving credit where credit is due. It's wonderful. Your caution about conclusions, because I believe it's from physics. There's nothing so sad as a theory destroyed by facts. and we all really want certain things to be true, but data don't care. And they never have. I mean, the truth that we find out in the lab, there's very little truth in the world, but there's a lot of truth on a petri dish if you look carefully. And so these are the things you've talked about. I love the inverse of what was expected and how you and your group responded to that. And people are going to have opportunities to read some of these papers and think about them. I have to really think about adding lots of extra links to this one, because this has been a fun
I'm going to
session
send you the wall metaphor.
Oh, sure.
Yeah, because, and I want our readers to think, do the words that we use limit the science that we do?
And that's just like, we
really
talk about
important.
this in everyday life, right? Yeah. And why wouldn't it be in science? And the number of times I've been told, for example, that my bacteria, and usually people I talk to don't discriminate between bacteria and archaea, but we'll leave that. And they'll say they don't have organelles. And I'll look at them. Well, they have to have different areas where things take place.
Exactly. The cytoplasm is not homogeneous.
It's not a big bag of enzymes.
That's right.
Right? That there's structure. I used to make a joke that membranes are like the Tupperware of life, because you have to separate things, and you really do. And so this is a good thing to think about. Arturo, it is always such a pleasure to see you.
It's a pleasure to be here. Thank you for having me, Mark.
Thanks again. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with magnificently epic web links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Arturo Casadevall is chair of molecular microbiology and immunology at Johns Hopkins University in Baltimore, Maryland. Many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum today. See you next time on Matters Microbial.